froze. So we heard about the place cells, grid cells, and head direction cells. And we've been studying these neurons in bats, in Egyptian fruit bats in particular. And we found over the years 3D place cells. We've been focusing in the early years on representation of 3D space. So Michael Yartsev, Arsene Finkelstein, and Gilles Guinosar, 3 PhD students in the lab, found 3D place cells, 3D head direction cells, and 3D grid cells. And 3D head direction cells, for example, were not on a spherical coordinate system, an toroidal coordinate system. And 3D grid cells, we found that they actually do not form a periodic lattice, two-dimensional lattice like FCC or HCP, but there is a local distance metric between nearby activity fields. So this was already all published, and I actually today I'm not going to talk about representation of 3D space, but about the representation of very large spaces in the brain. And in fact, I'll tell you today 2 stories, or I'll try to. One is sparse-to-dense coding transformation between hippocampal subregions CA3 and CA1. That's a study done in a 200-meter tunnel, so like a one-dimensional setup. And the other is the dimensionality of neural manifolds in the hippocampus of bats navigating outdoors on a remote oceanic island. So by way of background on the first part, some years ago, Tamir Eliav and Shir Maimon, two PhD students in the lab, have looked at presentation of very large spaces in CA1, in hippocampus CA1. The rationale was that bats navigate hundreds of meters underground and many kilometers outdoors. And so we'd like really to understand beyond the small laboratory setups, how is really large spaces represented in the brain. So for that, we developed a wireless neural loggers that allow storing the data on board the animal. And you can get really nice spikes in flight in the bats this way, as you can see here. And we've built a large behavioral setup, this long tunnel, 200-meter long. You just see the end of it. It's actually longer than that. And this was one of the first neurons that Tamir recorded early on. And as you can see, this neuron actually does not have this classical single Gaussian place field, as we typically tend to think about place cells in small environments, like in the small boxes, but it has multiple place fields, and they actually differ from each other in size. So here's a neuron with a large place field here, and a smaller place field here or here. And here's another neuron with a large place field here, a small place field here, a small place field here, a medium place field here. So sort of a multi-field, multi-scale representation of space in CA1. Looks very different from place cells recorded in small laboratory setups. And this is sort of summarizing the population summary of this. Many neurons have sometimes 10 or even more fields per direction, and they differed a lot in size from each other. And we've done, together with Yonatan Khadef, we've done some theoretical analysis of sort of what is the advantage of such a multi-field, multi-scale type of code. We compared 6 or 8 different types of neural codes and showed that for large environments, this type of neural code is much better than the single place field classic code. I'll show you a little bit of that in a second. Now this left an open question, which is what I'm going to want to show you now. And this was the focus of this project on sparse coding in CA3 versus dense coding in CA1. Worked by Shir Maimon, sort of together with Tamir, just the reversed order. And this was the important part of the CRCNS grant together with Yonatan Khadef and Lior Alas. Okay, so the basic question that we asked is, how does spatial coding differ, or does spatial coding differ in CA3 and CA1 large environments? So the basic sort of problem is that, or at least for my mind, it was a problem that for many years, people have been studying place cells in CA1 and in CA3, and CA3 project to CA1, so these are consecutive brain areas. Their anatomical connectivity is completely different. CA3 is highly recurrent, CA1 is much less so, and so you'd expect them something to differ, but in fact, people have not found big differences. There were some small differences people found over the years, but considering that these are completely different two different brain areas, you'd expect much bigger differences. That was not the case. So then the question that we posed is, maybe that's because these experiments were done in small environments. If you go to a larger, more naturalistic scenario, these brain areas will actually differ much more. So for that, Shir recorded simultaneously in CA1 and CA3 in bats flying in the same tunnel. And this is what she found. So here are 3 examples. Neurons in CA1, multiple fields as before, but in CA3, single fields. So completely different picture in simultaneously recorded neurons. And across the population, this is on a logarithmic scale, almost 80% of the neurons in CA1 and CA3 had single fields, as I just showed you. In CA1, we had this very long tail with many fields. So very, very big difference. However, the field sizes were in fact the same, sort of log normal distribution that did not differ between the two brain areas. So really in CA3, it's single fields that across neurons are distributed log normally. In CA1, it's the same thing, just with multiple fields. And if you now vary the environment size, 6 meters, 15 meters, 130, 180, and 200 meters, I want you to pause for a second to appreciate the amount of work that went into this. Then you see that the field sizes go up together, really match very nicely between the two brain areas. But the number of place fields increases in CA1, but does not increase in CA3. Okay, so we have a completely different type of neural code. Now that is a problem, because I just told you that this having multiple fields is better, and much better than having single fields. But CA3 has single fields, so that's pretty crappy neural code, according to what I told you a few slides ago. And just to show you, to drive home the message, this is median decoding error as a function of environment size in simulations with these two different neural codes, single fields or multiple fields. And in CA3, it shoots up. CA1, it doesn't. These are catastrophic, probability of catastrophic errors larger than 5% of the environment size. This is how many neurons do you need in order to represent the environment or to encode the position of the bat in the environment with a certain precision, let's say 2 meters. And you see that this big difference. So CA3 is really crappy in this respect. So that's odd. Why would you then have such an arrangement? And so here we took inspiration from work in the birdsong. So what I just told you might, for those of you who know a little bit about birdsong, ring a bell, because right, we have in CA3, we have a sparse code, in CA1, a dense code. And that's very similar to what happens in the birdsong, where neurons fire once within a song, just like in CA3, the neurons have one place field within the tunnel. And then downstream in area RA in the birdsong, neurons fire multiple times along the birdsong, just like in CA1, they fire multiple times. So we have the same architecture of a sparse coding area projected to a dense coding area. And Ila Fit, when she was a PhD student, did a model that tried to explain the utility of this type of modeling. And she showed that this is much better for faster learning. The intuition being that if you want to learn a target song in RA, then you want to modify a particular synapse at a particular location. Then if you have only one field, let's say place field in HVC, then you modify just one synapse, like a one-to-one modification. Whereas if you had multiple fields in HVC, then you'll want to change here, but you'll also inadvertently change in other locations. So create interference. So inspired by this, Yonatan Al-Khadef did a model that is similar to this for the case of the hippocampus, and found that indeed the learning time is much faster for one field in CA3 versus multiple fields in CA3. So this sparse-to-dense architecture allows faster and more accurate map learning. So the summary of this is that CA1, which is a major output of the hippocampus, shows this dense coding, this multiple field, which is good for precise coding. CA3 is not the output of the hippocampus, so we don't care that its coding sucks, but it shows sparse coding, which is good for fast learning. Okay, so that's sort of the summary. And we basically have a reformatting of information between CA3 to CA1 for better coding with less neurons. So CA1 shows this compressed output with a dense coding, and CA3 you can think of as a hidden layer in the middle of the hippocampus that has this code that's not good in terms of coding, but allows faster learning. And the one prediction of that, or consequence of that, is that if we make a local manipulation, then CA1 should be sensitive to local perturbations, while CA3 should not. So we actually did the experiment, moved one of the many landmarks in the tunnel, and showed that in CA1 neurons responded to this local change, and in CA3 they didn't. For lack of time, I won't show you this, but something that we've done. And for further questions in the model, you're welcome to talk to Yonatan Khadef, who is here. And we also have a poster on this at SFN, so you're welcome to come see. There's many more details here. Okay, in the remaining few minutes, I want to tell you a little bit about the work outdoors. So the basic question here was neural coding in the brain's navigation circuit during natural navigation outdoors. This was a long-standing dream of mine. And so basically what happens when you go to a situation on the left, like this tunnel, to the situation on the right, like outdoors. And this is a work done by Shaked Palgi, a PhD student in the lab, and CA3, a postdoc. So skip a lot of details. We've done electrophysiological recordings outdoors in bats flying on a remote oceanic island in Africa, in Tanzania, near Zanzibar, 70 kilometers off the coast of Zanzibar. The idea was that they cannot escape there. So this solves the problem of how do we recapture the animal. It's also a small island, so we get a very good coverage. And so this is how this looks during daytime. This is how we turn tetrodes during daytime. These are the tents where we've been sleeping for a month. This is the sea. This is me in Psycha turning tetrodes. You might see some clusters here. This is night starting to come. So this is my frisbee. And this is at night when you run the experiment. This is how the experiment looks. Okay, so we've done some work on head erection cells. We recorded in the dorsal presubiculum. So sort of a classical head erection area in 6 bats. I'm just showing you the trajectories of 2 of the bats here on the island. And we found 3 main findings. I'm just running quickly through them because I want to get to the manifold, which is the unpublished stuff. This was just published a few weeks ago. So the first finding is that there are head erection cells outdoors. You see a beautiful example here of a nicely tuned head erection cell, stable over time. And here are some more examples of head erection cells. You do dimensionality reduction, like with UMAP, you get a ring manifold because, of course, these are neurons that are tuned to a cyclical variable. So, of course, you'll get a ring manifold. The second finding was in terms of the stability of the compass over geographical space of the island. We found that it's stable. So here are examples of 2 neurons of the neuron in different spatial bins. So, for example, this neuron on the right prefers the green direction, the southeast direction, throughout the geographical extent of the island. This is a scale bar of 50 meters. And the neuron on the left prefers the red direction, the north direction, also throughout the geographical extent of the island. So the color uniformity of these maps mean that this neuron shows the same preferred direction everywhere. And we quantify this in many ways. This is just to show that the neurons are really stable geographically across the island, as you'd expect from a compass. And the second question, and the third finding was relating to celestial cues, because the moon is moving, the stars are moving, moon is setting, rising. So all these dynamics, if these neurons were sort of following that would be a problem. And so we did recordings of the same neurons without moon and with moon. Here are examples of 2 neurons. You see they're rock solid with moon and without moon. We also compared with cloud coverage, so including the moon and the stars, and it was rock solid throughout. Again, I'm just showing you examples, but we have very detailed quantification that you can go and have a look in this paper. So the bottom line is that there is stability of directional tuning over geographical space, and there is stability of the directional tuning despite movements of the moon and stars, suggesting that head erection cells outdoors can actually serve as a veritable neural compass. Now, this was all head erection cells. Now, what about place cells? So we did record place cells, as you can imagine, even though this is unpublished yet. So here's an example of a nice place cell that has 1 place field. We had other neurons that had multiple place field. And again, for lack of time, I'm not showing you all the details. We have a poster about this at SFN, so you can go see all the details about what the individual neurons are doing. But what I'm going to show you now is what the neurons is doing as a population, because in fact we've recorded quite a lot of neurons simultaneously, between 100 and 200 neurons simultaneously. So this is how for the aficionados, how the clusters look in 1 tetrode, and we have 16 tetrodes like that. So we had 4 bats with amazing recordings between 100 and 200 neurons. So we can now look at neural populations during natural behaviors outdoors. And so in particular, we looked at the dimensionality of the neural population manifolds. So basically, as you all know, you can use the n-dimensional population vector to construct the neural manifold, which is the collection of the dots of an n-dimensional space where n is the number of neurons. And many studies in rodents performing simple laboratory tasks report a low dimensionality of the neural manifold across different brain areas. And then, in fact, two types of dimensionality that people have been looking at: intrinsic dimension and embedding dimension. Intuitively, intrinsic dimension, for example, for this ring is 1, because it's, you can use one parameter to parameterize it. Also here, for the ring is 1, but the embedding dimension here is 2 because it sits in two-dimensional sheet, whereas here it's 3 because it's sort of folded. And Yuval Wasserman, a PhD student, joined with Misha Tzodix, has looked at these neural manifolds, and it found these dimensionalities, and it found that, and compared recordings outdoors to recording indoors, because we recorded the same neurons indoors in a bat flying in a tent, and then released them immediately a few minutes later outdoors. So we can compare the exact same 100 to 200 neurons indoors versus outdoors. And the result is that you see that the intrinsic dimension, each line here is a session, goes up by 2-fold, and the embedding dimension, that's a participation ratio for those who know about this, goes up in some cases 4-fold, so from 5 to 20, more than 20. Okay, so neural dimensionality, and importantly, neural dimensionality indoors is similar to reports in rodents. So if you look at the indoors values, it's very similar to what people have found indoors in mice, but yeah, I'm finishing. The dimensionality outdoors increased 2-fold for the intrinsic dimension and up to 4-fold for the embedding dimension. That's a very big effect. And the interpretation is that the dimensionality outdoors is so high because there's much more that these neurons are presenting because of the complexity of the environment, of the behavior, and of cognition. And in fact, you can subsample the number of neurons, and you see that the number of, as you sort of look at the session with 100-something neurons, then some sound to 90 or 60 or 30 neurons, and you see that the indoors, the dimensionality saturates. You know, you can record, look at more neurons, the dimensionality does not grow, but for the outdoors data, it keeps growing. Okay, so the conclusion from that is the low dimensionality in lab experiments that people have been reporting across brain areas could be an artifact of two simple tasks, and that to reveal the true complex structure of neural activity, it is not enough to only invest in increasing the number of neurons. Instead, brain research should combine many neurons with rich naturalistic behavior. Then you can get the full dimensionality of the manifold. Right, with this, of course, this makes me very happy because some of you know, I also published a book earlier this year called Natural Neuroscience, talking about why we should record the brain closer to the natural behavior of animal and humans. You're welcome to check it out. And with this, I'd like to thank you for listening.
Okay, we have a couple minutes for questions and then lunch.
Did the dimensionality evolve over time when you lived outside, or were they well trained in that environment already? So you were just looking at kind of like a learned snapshot?
So this is something we're looking at now. We don't have a complete answer here because, in fact, the bats were released there from day one. So the bats were from the mainland. They don't live on the island. From the mainland, I caught them on the mainland, then we brought them to this novel environment. So in fact, we can look from the very first millisecond what's happening outdoors. And to look like in some of the bats, there is some dynamics over days. In one of the bats, there isn't. So, you know, we're still looking at that, but it's definitely something we want to look at. Yeah.
Your plots there, the last slide, the intrinsic dimensionality starts off larger than the embedding.
Yeah. Right, because the question is how come, and you're right, there's no way this can be higher than that, right? Sorry, the other way around, this can be higher than that, right? You expect the intrinsic dimension to be lower than embedding dimension. The reason is that these are a little bit the very different metrics. This is computed through the correlation dimension. This is participation ratio, which underestimates it. If you'd use 80% of the explained variance species, it will be higher. So it's a bit of apples and oranges. Therefore, comparing these numbers directly is tricky. What you can really compare is, you know, within each of these metrics, you know, across the number of cells or between the indoors and outdoors, because that is exactly the same metric. But comparing the embedding to the intrinsic at face value is tricky. But you're right, it should be higher.
Okay, looks like you're all done. So let's do lunch.
Good.